I can probably hear many of you groaning internally, oh no, a nutrition talk, lots of numbers. I tried to minimize the numbers on the screen in that, but now I think I got carried away with a little bit too much text. So, but as we go through the presentation in that, feel free to go ahead and ask questions, or we can have a question and answer session at the end. And so, as Bill indicated, he asked me quite a long time ago if I'd be willing to give a talk on distillers, grains, and solubles. And to give you a little bit of background, at the University of Minnesota, I have an extension and research appointment. And most of my work is in the area of turkeys, market turkeys in particular. We used to do quite a bit of breeder research on the farm, but over time that got to be so expensive that we really couldn't find anybody that would fund us, and that's kind of gone by the wayside. But unfortunately, as time goes on, it's actually a much greater need than I think what a lot of people realize. So, but anyways, we started actually about probably about 10, 11 years ago now on DDGS. And it was kind of funny when we started out, people were really cautious about the product and using the product anyways. And, you know, if we kind of got people up to 5% in turkey diets, you know, they felt comfortable with that. And then over time, we did more and more. And then 2008, 2009 with high feed ingredient prices, and now this is with market turkeys. We actually had, you know, some people in the field that were actually going up to 20 to 25%. With our research, we actually found with market birds, depending upon the age in that, we could go up as high as 30% DDGS in the diets. A lot of caution, though, when you go to using those high levels, and we'll talk a little bit more about that. So anyways, that's my background. I'm a turkey person, and as you all realize, you know, 44-pound turkeys are quite a bit different than the size of the bird that you're working with. So. Okay, so what I'd like to do during the presentation is just first talk in general about DDGS, what is it, and as we go through the talk, we'll talk a little bit more about how it's changed over time. And then I'm going to concentrate more from the standpoint of, since some of the background I had gotten indicated that, you know, there were concerns about using the DDGS in pheasant breeder diets, and so I'm going to emphasize a little bit more on the reproductive side for that. Talk a little bit more about some precautions, you know, we've developed over time in terms of feeding the material to the birds, and then some general recommendations for use at the end. Okay, so what is DDGS? The initials anyways stand for dried distillers, grains with solubles, and basically that's the form that it will typically be fed in poultry. In swine, there's some different modifications of that product for the ruminant animal. Over time, many years ago, in a lot of the initial early research in the 60s and 70s, was actually involved with distillers grains that were the result of some kind of beverage alcohol fermentation. So whether you're producing beer, in which case you'd have brewer's dried grains, or if you're looking at some of the whiskey distillers, you would also could get a distiller's dried grains with solubles product from that as well. We have, as indicated this morning, it appears to be a really diverse attendance here at the meeting. The UK, China, so forth, and also people out on the West Coast. Well, we're in the Midwest here, so most of the distillers grains we're going to be working with is going to be corn. And so really, when you're working with that product anyways, you have to know what the basis of the grain source is for the fermentation, because that's going to determine a lot in terms of what kind of nutrient composition that has. And so as I go through my presentation this morning, keep in mind I'm just talking strictly corn distillers grains here, not wheat, not barley, and so forth. Okay, so just a little bit of an overview here with, give you a little bit of background here in terms of how distillers grains is produced, and this is more the traditional method. And like any industry, the ethanol industry is challenged with trying to increase efficiencies in that, and they're also looking for obtaining some added value for the products that they're producing at the end besides the ethanol. And so lots of different changes, lots of different innovations, developments going on almost on a constant basis. And so kind of keep in mind this is the picture for, you know, maybe a product that might have been produced 5 to 6 years ago. And we still have some of that product being produced, but with lots of different modifications in that. So basically, in the ethanol plant then, the corn will come in. Hopefully it's undergone some screening and testing for quality in that. Basically, the next process then is to take and set that corn up for the fermentation process. And then after the material is fermented, you'll have the ethanol that's going to be taken and distilled off. And then you'll have what's called the stillage. And so this is what's ever remaining after the fermentation process has been completed. So basically the fermentation process is going to use primarily the starch and the sugars that's present. They may add some enzymes in there as well to try to take and break down some of the fibers too and get some material, some energy for the yeast cells. And then we also have the yeast that will be part of this whole stillage as well, the dead yeast cells anyways. And then basically end up with kind of two product streams, if you want to call it that. One is that's called the thin stillage, which basically is called the syrup, and that's going to be quite a bit of the corn oil. It's got some particulates in it. It also contains a fair amount of phosphorus in it as well. And then you'll have the more coarse wet grains material. And then, so anyways, you go through some initial evaporation. That's going to be added back to the solids in order to get the distiller's dried grains with solubles, and then that will be dried in some kind of dryer. And so you'll have your DDGS product there. If you didn't want to add that syrup back, or you only wanted to add a portion of it, and if there was a market for the distiller's solubles itself, you know, that could be taken and dried and give you a product called condensed distiller solubles. And then, as I said, in the ruminant market anyways, they can use some of the distiller's grains that haven't been dried or not dried completely. But again, for poultry and swine, we're going to be looking at a dried product with the syrup, at least some portion of the syrup added back. Okay, so why would we take and add distiller's dried grains to poultry diets then? And primarily as a source of energy. The conventional product has a fair amount of residual corn oil left in it. As a source of protein and amino acids, and we'll talk a little bit more about some of the limitations there as well. And then also as a source of phosphorus. Cost of phosphorus has really increased significantly here in the last few years, and that's actually made the DDGS product a little more valuable. In terms of when is it going to come in, it really all, besides the nutrient contributions, it's really all going to depend upon what's the price of corn, soybean meal, because we're using it as a source of energy and protein. And then also what's the price of some of the supplemental amino acids, because DDGS is very deficient in lysine, and so that deficiency needs to be made up. And then depending upon the type of product, then you may actually need to add back a little bit of fat or oil to bring the energy level up, because energy level of DDGS is about 85% of corn. And then, like I said too, you know, what's the price of your phosphorus sources? So in this case, I just used the example of dicalcium phosphate. Okay, well, I don't see too many people sleeping in the back there yet, so. Okay, you know, we kind of went through this, you know, back in the 60s, 70s, 80s, and then maybe into the 90s, where we talked about what the conventional DDGS was, and it did tend to be really a byproduct. Nobody paid much attention to it. They were just interested in getting the ethanol out and then getting that product sold in one way or another. So not a lot of quality concerns relative to using it as a feed, and lots of people had lots of suspicions about even incorporating it into the diet. And then as more ethanol plants came online with the renewable fuels incentive, then we started calling it the new generation DDGS. And that was because, again, developments in the ethanol processing and you know, people were actually starting to look at it a little bit more as a feed ingredient, what they were going to do there to try to take and improve the quality of it. So the new generation anyways, somewhere is around 26 to 28% crude protein, about 10 to 12% fat, and that would be as corn oil, and then somewhere is around 0.7 to 0.75% phosphorus. Okay, so what's happening now? Basically, you know, if you talk to the ethanol guys, they'll talk a lot about fractionation, things that can be done at the front end of the processing, things that can be done at the back end of the processing to take and basically fractionate the corn into a lot of different products. So they can take out the fiber, they can isolate the protein, they can take out the oil. And so now, instead of having this new generation DDGS product that we're talking about, now we have multiple different types of corn co-products that can be available. And even at a very basic for the new generation product, one of the things we immediately noticed was depending upon how much of the syrup the ethanol plant would actually add back to the wet grains and dry it, that has a huge impact on the fat level of the product. And so, from an ethanol plant's mind frame is, okay, if we can sell these solubles someplace else, we don't have to spend the money to take and dry that product down, or as much energy to take and dry it down. So, they're kind of, a lot of what you'll see at the ethanol plants is a lot of times they don't have a good understanding of what is needed to have a high quality feed ingredient. They're making decisions based on the economics of running the ethanol plant. Mentioned already about corn fractionation. One thing that is really coming on very fast right now is something that they call spinning. And basically all they're doing is we've got that syrup that has a high content of oil. They're taking and removing that oil out of the syrup, or a portion of it anyways, and then adding the remaining syrup back to the wet grains to take and dry that. And I think for 2012 it was, or 2011 it was estimated about 50% of the ethanol plants were doing this. And by the end of 2013, I think they're estimating 75% of the plants will be producing this. It's what we call a reduced oil product. And we'll talk about some of the implications about that later on. So anyways, then we also have plants that will take and remove, actually they will, remove the oil, take out some of the fiber, take out some of the phosphorus and so forth, and have a very high protein product that will be somewhere, 36, 38, all the way up to 50% crude protein. And then as I mentioned, 75% of all plants by the end of 2012 will be producing a reduced oil DDGS. So if you kind of remember what I talked about, the new generation DDGS was about 10 to 12% oil. Now we're going to have products that will range somewhere around 6 to 8% oil. Okay, so anybody that's worked at DDGS has heard about variability, and so you have variability of lots within a plant. You have variability, quite a bit of variability then of material among plants, just because of all the different processing techniques and so forth that are used. And can we get the lights? Front there. Okay, thanks. Can't really see this a whole lot, but if you actually had this sitting in a tray in front of you or something, you could see some very distinct differences just based on the color and the particle size and that of different types of DDGS. Depending upon the system, like Poet, they basically have tried to standardize all their plants to produce a more standard product, and so that helps with some of that material. But other places, everybody's kind of trying to do something different, as I mentioned. So we talk about variability. You also kind of have to keep in the back of your mind that many other ingredients have variability too. Corn from location in the state year to year is going to vary. Soybean meal is going to vary. Any other byproducts that you might be using in the feed are also going to vary. But so just to kind of keep, not to pick on DDGS per se as being a culprit here. So some data from Jerry Shearson. Again, this would be more the new generation type product. And the values in the parentheses here indicate what we call the coefficient of variation. And a smaller number means that that product or that particular measure is much more uniform. And what we'd ideally like to see would be something that would be close to 5%. That's really hard to reach. So something in the range of 5 to 10% would be really great. And so, for the most part, what you can see, and this is all on a dry matter basis now, getting the dry matter crude protein usually isn't an issue. Now we start to see a little bit more variation in the crude fat levels. Fiber just has a lot of variation with it just because it's I shouldn't say difficult to measure, but there's variability associated with that. Things that we're really concerned about here then, besides the crude fat, would be the lysine level and the phosphorus level. And so again, you can see, a pretty huge amount of variability for those two really key critical nutrients for poultry. And we haven't even talked about the digestibility of those two nutrients for the birds as well, which adds an additional layer of variability. So what we're doing now is we've got to worry about variability within plants, among plants, and then we're going to throw on top of that we might see variability because of different co-products being put into production chain, but not knowing that that different co-product is there, okay? So you might have expected that you were purchasing a new generation DDGS, and you might end up with a reduced oil DDGS instead, or a high protein DDGS. Okay, so lots of research. As I mentioned, we've done quite a bit of work at the University of Minnesota for turkeys, and University of Georgia, various places, Illinois and so forth, have done a lot of work on broilers and chicken layers as well. And so we've got a pretty good database of what's going on, at least with the new generation product. And kind of getting back to my opening statement, the type of grain source used in fermentation is really sets the basis for what kind of nutrient composition you're going to have. And in chicken layers anyways, we like to talk about the contribution of the pigment xanthophyll, the orange-yellow type pigment, and that has some value there in chicken layer diets for production of eggs for human consumption. We know that processing can take and affect the nutrient value, and basically anytime you take and heat something, and if you have a combination of some kind of carbohydrate along with amino acids, particularly lysine, we get something that's called heat damage. And all that is a complex that's formed between the amino acid and the sugar, but it's not digested by the bird. And so we pay a lot of attention to lysine digestibility on DDGS because of there's lots of heat applied during the fermentation, drying, distillation, and so forth. Likewise, opposite is the availability of phosphorus. Actually, the availability of phosphorus of the bird, we have, it's called a phytate phosphorus molecule that birds do not have the enzyme to take and digest that. And so actually kind of opposite to lysine, when the DDGS undergoes heating, it actually tends to release more of that phosphorus from the phytate molecule. And so we actually have a higher availability anyways for phosphorus so that the bird can take and utilize more of that and excrete less. And I think we have, usage levels fairly well defined for commercial chicken and turkey, but probably for many of the other species in that, and breeders in particular, we don't have as good a guidance there. Okay, I'm going to have to really start moving here. I was thinking when Ron got done about 10 minutes earlier, that would give me 10 more minutes to talk, but then there was lots of good questions and answers. So to go through chicken layer hens anyways, and basically what this says is the maximum level of DDGS that was fed, what the impacts were, how the diets were formulated, and who the reference was on this. And basically, just to pick out a couple of things, 15% distillers and chicken layers, especially when the diets were formulated on what we call a total amino acid basis instead of a digestible amino acid, and also contained slightly lower nutrient levels anyways, that they did have some problems here with the birds in terms of some decreased egg production and increased feed per egg laid. Roberson actually noticed initially at the start of the trial that there was a decrease in some of the production parameters. And then paper out of University of Nebraska, they actually started to see a negative effect of DDGS inclusion at 15% that could take and be corrected with some amino acid supplementation. And then surprisingly, a really short-term trial for 4 weeks, they actually fed up to 69% distillers grains in a chicken layer diet, which was even beyond what I would ever think that we could take and put in. And they basically only had a negative effect at the very highest level that they fed at the 69%. But again, this was over a 4-week time period. And actually, Dr. Stacy Roberts, who will be talking, I think, on Tuesday or Wednesday, she was actually, I think, part of this trial too. If we look at breeding poultry anyways, really limited data there. 3% in turkey breeders, no negative effects, but the performance of the controls are really poor, and so then that kind of makes you question the trial. 10% chicken in chicken leghorn diets, no effect at 5%, but they did see some decreased hatchability and fertility. And that really, again, comes back to this amino acid lysine. And so once they put that lysine in the diet, restored the reproductive performance back to normal. Personal communication, with broiler breeders anyways, that they're including up to 30% in their rearing diets as well as in their broiler breeder diets, and not seeing any negative effects at all. But you kind of have to keep in mind a broiler breeder diet and a much larger growing bird, larger capacity for feed intake, even though they will be restricted fed for most of their life anyways. But the main point there the nutritionist made is that you really need, again, to keep an eye on the energy and the digestible amino acid levels. Kind of going back and forth with Bill and Brad anyways, you know, they feel that, or that they are seeing negative impacts by including distillers grains in terms of egg production, fertility, and also impacts on the health and the mortality level of the progeny. And so this kind of put me in a little bit of a conundrum, as they say, because I was looking at it that there's lots of research-based data anyways out there that says, you know, you should be able to feed to chicken layers anyways about 10 to 15% DDGS. And so for me then is I had to kind of reshape my thinking a little bit and think about it, okay, from the standpoint of, you know, what might be causing some of the poorer performance with pheasant breeders. So kind of just a list of things here. What's the difference, research versus field? At times, we can do a lot more in a research setting that they can do in the field, more controlled studies and all of that. And so it's probably not surprising, you know, that you might not be able to come in and feed really high, what we could feed in a research setting. Difference in species, bird size, feed intake levels, and so forth. Might be some issues with proper product identification for the distillers, issues of product quality, maybe some other excessive nutrients or contaminants that might be present, such as differences in balance of electrolytes or presence of mycotoxins. So to kind of go through corn co-product composition here, I know I was trying to cut down on numbers here, but I still ended up with a fair amount anyways. But so if we have our new generation DDGS, a reduced oil DDGS, a reduced oil here that really has very minimal crude fat levels back, and then a high protein DDG. And so what you can see is, you know, we are going to see a definite trend in terms of reduced oil content in these DDG products. That in turn takes and affects the metabolizable energy content of those products. When protein starts to get concentrated, we start to see a little bit more lysine present in the diet, and that's a good thing. But we have to keep in mind, you know, what the digestibilities of those amino acids are. And as I mentioned, phosphorus, we have a big switch here. I don't have values for these reduced oil products. I'm assuming that they'll be a little bit higher than what we have here. But you can see with a high protein DDG, 42% crude protein, you know, fairly large decrease in the amount of phosphorus that would be present in that product. So if we just took those and assumed we were going to throw them into the diet at 20%, not make any nutrient adjustments, then what we have is about 20 kcal per kilogram decrease. It's not a whole huge amount. It would be like around a little bit over 0.5%. In this case, with this product, it's a huge decrease in energy. Digestible lysine actually, because of the increases in the lysine content, we actually end up with an excess of digestible lysine, which is costly because you're already adding lysine to try to decrease it or correct a deficiency. Available phosphorus as well, we could end up potentially with some excesses with the high protein. We actually end up with a deficit in phosphorus level. So to kind of really summarize this very quickly, metabolizable energy anyways, birds eat to meet their metabolizable energy need. So if they perceive a shortage relative to the requirement, they're going to increase their feed intake. The big problem issue here is, do they have the capacity to take and increase their feed intake? If they're already eating maximally and you give them a lower energy diet, you know, they just may not be able to take and make that correction. With a shortage of lysine, we know we would get a decreased egg weight and egg production. When we have an excess, it actually creates amino acid balance, which can also take and indirectly affect feed intake then. Phosphorus issues, either with the shortage or excess, we can get into some shell quality issues. and likewise with the electrolytes, we can get into some shell quality issues. No, mycotoxins, particularly when you're talking about it from a reproductive standpoint and as compared to meat birds, I know there's different issues, but the risk is probably definitely higher, for breeding birds anyways. And again, in general, for corn in the Midwest, we typically think of relatively low levels of mycotoxin contamination, but this is going to vary by year to year. One thing that our state climatologist is really starting to try to get people to recognize is that we are seeing a lot more variability in weather patterns during the summer. I mean, in Minnesota, we had some really heavy rainfalls at first, and I think we didn't have any rain for the rest of August. Rainfall is going to be much more spotty, and so chances are some fields will get adequate rainfall, some fields are not going to get adequate rainfall. So I think, we need to keep this in mind for the future. As I mentioned, ethanol plants should be screening for mycotoxins. They do claim anyways that, they want a good quality corn to come in because that helps with the efficiency of ethanol production. The thing to keep in mind, at least for what we call the new generation product anyways, and the reduced oil, is that if there are mycotoxins present in the corn, they'll be concentrated by a factor of about 3 because the fermentation and so forth and heating does not take and destroy those toxins. Toxins that we would associate with fungal infections are the grains, aflatoxin, deoxynivalenol, DON, I hate pronouncing these names, fumonisin, T2 toxin, ochratoxin, and zearalenone. Part of the issue is if you want to find something, you have to do a lot of tests, and that can get to be costly over time. Mycotoxins, at least until recently, there weren't a lot of accurate, rapid testing available. And then you also end up in this interpretation of results because a lot of times what you see in terms of research results versus what happens in field outbreaks is there's a huge difference in terms of the levels of what we consider to be performance, causing negative effects on performance or actually causing toxicity. But in general, poultry performance, we're talking about, numerous effects: feed intake, reduced egg production, hatchability, shell quality, reductions in immune response, increases in embryo abnormalities, and decreases in progeny performance. As far as I can tell, doing some reading, it seems like pheasants are kind of intermediate, chickens being the most resistant, and ducks being the most sensitive in terms of effects on some of these mycotoxins. So one survey that was done, and I tried to summarize here, we have the aflatoxin levels, DON, fumonisins, and T2 toxin. We have what's called the lowest FDA either action level, advisory, or guidance level here. And then what these different surveys anyways found over time. And basically you can see that the levels of aflatoxin, and again, this is really going to vary from year to year, were very low. DON was relatively low. Fumonisins popped up a little bit more here. And no T2 toxins were actually found, at least in 2 of the subsets. But the thing to keep in mind anyways is that across these 3 surveys anyways, none of the samples exceeded the lowest action level for FDA for aflatoxin, which was 20 parts per billion. For DON, there were 0 that exceeded 5 parts per million, whereas for the fumonisins, there was about 10, 3, and 10% of the samples that were tested that exceeded the lowest FDA level, but still would have been considered to be acceptable to feed to poultry. So, if you do another year or with some different tests in that, you might see some different results. But it's one of those areas that you need to keep looking at. So, anyways, all of this and all of these, when we're talking about nutritional variability or mycotoxins, there needs to be a lot of communication going on between the ethanol supplier, the feed mill, or who's ever purchasing in the, in the farm, what's going on at the farm level. And so from the supplier anyways, get in contact with them and see, okay, what kind of a corn testing program do they have? Do they have a mycotoxin testing program for the corn and the co-products that they're producing? Are they communicating any process changes for the purchaser anyways? conduct mycotoxin testing. There are approved tests for DDGS, and you have to be careful because a lot of these tests, especially the rapid tests, have been developed for specific ingredients. So you need to use one that's been validated for DDGS. And there's a website on the government, GIPSA. If you just do GIPSA mycotoxin for a word search, you'll find that they have what they consider to be approved test kits. And there's always the issue of obtaining a representative sample. Likewise, you can keep track of what's going on locally with your corn crop. Some of the extension offices, state veterinary diagnostic labs may have some of this information available. Some of the commercial labs will actually take and maintain a website, so Dairyland is one of those. Issue here is that I think there's a couple of sites at Minnesota that report in Wisconsin, but you really don't know where the samples are coming from. And then as producers, to maintain a feed sampling and retention program, get some analyses done, make sure you communicate as quickly as possible to the feed mill any issues or performance problems that you're seeing. So I think, so the bottom line kind of, I think feeding some low levels, somewhere in the 5 to 10% level, you should be able to do that with pheasants. But a lot of it comes back to evaluating your risk versus feed cost savings, and then what kind of testing and product quality assurance is in place to make sure that you're getting a good quality feed.